Introduction to autoimmune limbic encephalitis, ALE. Autoimmune limbic encephalitis, ALE, is a significant neurological condition characterized by the inflammation of the limbic system, a complex set of structures deep within the brain that play a crucial role in emotions, memory, and behavior. This condition is primarily caused by the body's immune system mistakenly attacking healthy cells in the brain, particularly within the limbic system. The onset of ALE is often sudden, and can lead to a variety of neuropsychiatric symptoms, including memory disturbances, seizures, confusion, altered mental states, and psychiatric manifestations such as anxiety, depression, and hallucinations. ALE is a part of a broader category of autoimmune encephalitides, where the immune system targets neuronal surface antigens or intracellular proteins, leading to inflammation and damage. This autoimmune response can be triggered by a variety of factors, including infections, tumors, paraneoplastic syndromes, or it may occur spontaneously with no clear underlying cause. Understanding ALE is of paramount importance in medical research, because it not only provides insight into the mechanisms of autoimmune diseases, but also because of its direct impact on human health. ALE can be challenging to diagnose and treat, as its symptoms overlap with other neuropsychiatric disorders, and the underlying immune mechanisms are complex and not fully understood. This has driven the need for robust animal models that can replicate the human condition and allow researchers to study the disease's pathophysiology and test potential treatments. In the realm of autoimmune encephalitis, animal models have been invaluable. They provide a controlled environment in which the disease's progression can be studied in detail, and therapeutic interventions can be tested before being considered for human trials. Traditionally, rodent models have been the mainstay of neurological research, but they have limitations, particularly when it comes to complex brain structures like the limbic system. As a result, there has been growing interest in developing models using animals with more complex brain anatomy, such as cats, to better mimic human neurological diseases. This study's focus on developing a feline model of spontaneously occurring autoimmune limbic encephalitis represents a significant step forward. Cats share more anatomical and functional similarities with humans in their brain structure compared to rodents, making them an excellent model for studying neurological diseases that affect the limbic system. By studying a naturally occurring form of ALE in cats, researchers can gain insights into the disease's progression in a way that is more directly applicable to human conditions. The feline model described in this study is particularly valuable because it occurs spontaneously, without the need for artificial induction of the disease. This spontaneous occurrence more closely mirrors the human condition, where ALE can develop without any clear external triggers. By studying these naturally affected cats, researchers can observe the disease's natural progression, identify early biomarkers, and investigate the immune mechanisms at play. The study of autoimmune limbic encephalitis in cats is not just about understanding a disease in animals, but is a crucial step towards unraveling the complexities of this condition in humans. This research has the potential to lead to better diagnostic tools, more effective treatments, and ultimately, improved outcomes for patients suffering from ALE. The following sections will delve into the specific details of the feline model developed, the clinical presentations observed, and the broader implications for human medicine. Development of the feline model. The development of a feline model for spontaneously occurring autoimmune limbic encephalitis, ALE, represents a significant advancement in the field of neuroimmunology. This model provides a unique opportunity to study a naturally occurring neurological disorder that closely mimics human ALE. The process of developing this model involved a careful selection of subjects, extensive diagnostic evaluations, and a thorough understanding of the disease's pathology in both cats and humans. Selection process for the feline model. The initial step in developing the feline model was identifying cats that naturally exhibited symptoms consistent with autoimmune limbic encephalitis. Unlike rodent models, where the disease is often induced through genetic modification or exposure to specific antigens, the feline model relies on the spontaneous development of the condition. This is a crucial aspect of the model, as it more accurately reflects the unpredictable nature of ALE in humans, where the disease can arise without any obvious external triggers. Researchers focused on veterinary clinics and hospitals as primary sources for identifying potential subjects. Cats that presented with neurological symptoms, particularly those affecting behavior, memory, and motor function, were of particular interest. These symptoms included seizures, disorientation, altered consciousness, and changes in personality or behavior, which are hallmark signs of limbic system involvement. Cats exhibiting such symptoms were further evaluated to determine if they met the criteria for ALE. Diagnostic methods. Once potential subjects were identified, a series of diagnostic tests were conducted to confirm the presence of ALE. These tests were crucial in distinguishing ALE from other neurological disorders that can present with similar symptoms, such as infectious encephalitis, brain tumors, or metabolic disorders. Magnetic resonance imaging (MRI). One of the primary diagnostic tools used was MRI, which provides detailed images of the brain's structures. In cases of ALE, MRI typically reveals inflammation and swelling in the limbic system, particularly in the hippocampus and amygdala. These findings are similar to those observed in human patients with ALE, where the limbic system shows signs of inflammation and, in some cases, atrophy due to chronic inflammation. Cerebrospinal fluid (CSF) analysis. Analysis of the cerebrospinal fluid (CSF) was another critical diagnostic step. In ALE, the CSF often shows elevated levels of white blood cells, pleocytosis, and the presence of specific autoantibodies that target neuronal antigens. Identifying these autoantibodies is a key factor in diagnosing autoimmune encephalitis. In the feline model, CSF samples were analyzed for similar markers, and the presence of these autoantibodies provided strong evidence of an autoimmune process at work. Histopathological examination. Postmortem examinations of the brain tissue provided further confirmation of the diagnosis. Histopathological analysis involves examining brain tissue under a microscope to identify signs of inflammation, neuronal loss, and the presence of immune cells, such as lymphocytes and plasma cells, in the brain. These findings are characteristic of autoimmune encephalitis and were consistently observed in the affected feline subjects. Electroencephalography EEG. Although less commonly used in veterinary medicine compared to human neurology, EEG was employed in some cases to assess the electrical activity of the brain. Abnormal EEG patterns, such as focal slow-wave activity or epileptiform discharges, can support the diagnosis of limbic encephalitis. In the feline model, EEG findings were consistent with those seen in human ALE, further validating the model's relevance to human disease. Identification of naturally occurring ALE in cats. The naturally occurring aspect of ALE in cats is what makes this model particularly valuable. Unlike induced models, which may not fully capture the complexity of the disease, the spontaneous occurrence in cats offers a more accurate representation of the disease's development, progression, and response to treatment. In this model, researchers identified a small cohort of cats that met all the diagnostic criteria for ALE. These cats were observed over time to study the natural history of the disease. The progression of symptoms, response to treatment, and long-term outcomes were all carefully documented. This longitudinal approach provided insights into the disease's trajectory, including periods of remission and relapse, which are also seen in human patients with ALE. Relevance to human ALE. The development of the feline model was guided by the goal of creating a model that closely mimics human ALE. Cats were chosen for their anatomical and physiological similarities to humans, particularly in the structure and function of the limbic system. The similarities in brain structure between cats and humans mean that findings from the feline model are more likely to be translatable to human conditions than those from rodent models. Moreover, the spontaneous nature of ALE in these cats mirrors the unpredictability of the disease in humans. This aspect is critical because it allows researchers to study the disease as it occurs naturally, providing a more authentic understanding of how ALE develops and progresses over time. The feline model also allows for the exploration of genetic, environmental, and immunological factors that may contribute to the spontaneous development of ALE, which could lead to the identification of new risk factors or therapeutic targets in humans. The development of a feline model for autoimmune limbic encephalitis marks a significant advancement in the study of neuroimmunological diseases. By providing a naturally occurring model that closely resembles human ALE, this research opens new avenues for understanding the disease's pathophysiology and testing potential treatments. The following section will delve into the clinical presentation and pathology observed in this feline model. Clinical presentation and pathology. The clinical presentation and pathology of autoimmune limbic encephalitis, ALE, in the feline model offer crucial insights into the disease's manifestations and its impact on the brain. By studying these aspects in naturally affected cats, researchers can draw parallels with human ALE, advancing our understanding of the disease's progression and potential therapeutic interventions. Clinical symptoms in the feline model. The cats in the study exhibited a range of neurological and behavioral symptoms that closely resemble those seen in human patients with ALE. These symptoms were often the first indicators that a cat might be suffering from this autoimmune condition. The onset of symptoms varied among the subjects, with some cats displaying acute, rapidly progressing signs, while others showed a more insidious, chronic course. Seizures. One of the most common and prominent symptoms observed in the feline model was seizures. These seizures were typically focal, affecting specific parts of the brain, but could sometimes generalize to involve the entire brain. The focal seizures were often associated with the limbic system's involvement, manifesting as sudden, brief episodes of abnormal behavior, such as staring spells, unprovoked aggression, or compulsive movements. These episodes were analogous to the focal seizures observed in human ALE, where patients may experience similar behavioral abnormalities. Memory and cognitive impairment. Memory disturbances were another significant symptom in the affected cats. Owners often reported that their cats seemed disoriented or unable to recognize familiar people or objects. This type of memory impairment is characteristic of limbic system dysfunction, particularly involving the hippocampus, which plays a crucial role in memory formation and retrieval. In humans, similar memory issues are a hallmark of ALE, contributing to the condition's profound impact on daily life. Behavioral changes. Behavioral alterations were widespread among the feline subjects. These changes ranged from increased aggression or irritability to depression-like symptoms, such as withdrawal and loss of interest in usual activities. Some cats exhibited hyperactivity, restlessness, or anxiety, which could be attributed to the involvement of the amygdala, a key limbic structure responsible for emotional regulation. In human ALE, psychiatric symptoms like anxiety, depression, and even psychosis are common, reflecting the disease's deep impact on the limbic system's emotional centers. Altered consciousness. Several cats in the study experienced episodes of altered consciousness, where they appeared to be in a stupor or a daze. These episodes could last from a few minutes to several hours and were often followed by a period of confusion or disorientation. This altered consciousness is similar to what is seen in human patients with ALE, who may experience transient episodes of confusion, delirium, or even loss of consciousness during the disease's acute phases. Motor dysfunction. Some of the affected cats also showed signs of motor dysfunction, such as ataxia, loss of coordination, muscle weakness, or abnormal gait. These motor symptoms could result from the spread of inflammation beyond the limbic system to other parts of the brain, such as the cerebellum or motor cortex. In humans, motor deficits can occur in more severe cases of ALE, particularly if the disease spreads or if there is significant brain damage from prolonged seizures or chronic inflammation. Pathological findings in the feline model. The pathological examination of brain tissue from the affected cats revealed several key findings that are consistent with autoimmune encephalitis. These findings not only confirmed the diagnosis of ALE, but also provided insights into the underlying mechanisms of the disease. Inflammation of the limbic system. The hallmark of ALE is inflammation within the limbic system, and this was clearly observed in the feline model. Histopathological analysis of brain tissue showed significant inflammation in the hippocampus, amygdala, and other limbic structures. This inflammation was characterized by the presence of lymphocytes, particularly T cells, and plasma cells infiltrating the brain tissue. The inflammatory cells were often clustered around blood vessels, a pattern known as perivascular cuffing, which is indicative of an immune-mediated process. Neuronal loss and gliosis. Alongside inflammation, there was evidence of neuronal loss in the affected limbic structures. This loss was most pronounced in the hippocampus, where neurons were found to be shrunken and pycnotic, a sign of cell death. In response to this neuronal loss, the brain exhibited gliosis, a process where glial cells, such as astrocytes, proliferate in an attempt to repair the damage. Gliosis is a common response to brain injury and inflammation, and its presence in the feline model is consistent with findings in human ALE, where it is associated with chronic disease stages and contributes to long-term cognitive deficits. Demyelination. Some of the affected cats also showed evidence of demyelination, where the protective myelin sheath surrounding nerve fibers was damaged. This demyelination was primarily observed in the white matter adjacent to the inflamed limbic structures. Demyelination can disrupt the normal transmission of electrical signals in the brain, leading to various neurological symptoms, including those seen in ALE. While demyelination is more commonly associated with diseases like multiple sclerosis, its presence in ALE highlights the broad impact of the autoimmune response on the brain. Presence of autoantibodies. One of the defining features of autoimmune encephalitis is the presence of autoantibodies that target specific proteins in the brain. In the feline model, researchers identified autoantibodies that were directed against neuronal surface antigens, similar to those found in human patients with ALE. These autoantibodies were detected in both the blood and cerebrospinal fluid (CSF) of the affected cats. The identification of these autoantibodies provided further evidence that the disease in these cats was indeed autoimmune in nature and closely related to human ALE. Synaptic loss. In addition to neuronal loss, there was also evidence of synaptic loss in the affected limbic structures. Synapses are the connections between neurons that allow them to communicate, and their loss can lead to significant impairments in brain function. The synaptic loss observed in the feline model is particularly concerning because it may underlie some of the cognitive deficits, such as memory impairment, seen in these cats. In humans, synaptic loss in the limbic system is associated with severe cognitive decline and is a critical area of research for understanding and treating ALE. Comparison with human ALE. The clinical presentation and pathological findings in the feline model show striking similarities to those seen in human patients with ALE. The shared symptoms, such as seizures, memory impairment, and behavioral changes, underscore the relevance of the feline model to human disease. Moreover, the pathological features, including inflammation, neuronal loss, and the presence of autoantibodies, provide strong evidence that the disease processes in cats are analogous to those in humans. This close resemblance suggests that the feline model could be an invaluable tool for studying the mechanisms of ALE and testing potential therapies. By understanding how the disease develops and progresses in cats, researchers can gain insights that may be directly applicable to human ALE, potentially leading to better diagnostic markers and more effective treatments. The clinical and pathological findings in the feline model of ALE offer a detailed and accurate representation of the disease, making it a powerful model for advancing our understanding of autoimmune encephalitis. Immunological findings. The immunological aspects of autoimmune limbic encephalitis, ALE, are crucial to understanding the disease's pathogenesis and developing targeted treatments. In the feline model, researchers were particularly interested in the immune responses that drive the disease, especially the role of autoantibodies and immune cells in attacking the brain. The immunological findings from this model provide significant insights into how the immune system contributes to the development and progression of ALE, and these insights are highly relevant to human cases of the disease. Autoantibodies and their role in ALE. One of the most critical immunological findings in the feline model was the identification of autoantibodies targeting specific neuronal proteins. Autoantibodies are antibodies produced by the immune system that mistakenly recognize and bind to the body's own proteins, leading to an autoimmune response. In the context of ALE, these autoantibodies target proteins located on the surface of neurons, particularly in the limbic system, leading to inflammation and neuronal damage. Identification of specific autoantibodies. In the feline model, researchers detected autoantibodies in both the blood serum and cerebrospinal fluid (CSF) of affected cats. These autoantibodies were found to target neuronal surface antigens, similar to those identified in human patients with ALE. The most commonly targeted antigens in both the feline model and human cases include proteins like the N-methyl D-aspartate receptor, NMDAR, which plays a critical role in synaptic plasticity and memory function, and the leucine-rich glioma, inactivated 1-LGI1 protein, which is involved in synaptic transmission. The detection of these autoantibodies in the feline model was a significant finding, as it confirmed the autoimmune nature of the disease and provided a direct link between the immune system and the neurological symptoms observed. The presence of these autoantibodies was associated with the severity of the disease, with higher levels correlating with more pronounced symptoms and greater brain pathology. This correlation is also seen in human ALE, where autoantibody levels often reflect disease activity and can be used as a marker for diagnosis and treatment monitoring. Mechanisms of autoantibody-mediated damage. The autoantibodies identified in the feline model contribute to the disease by binding to their target antigens on neurons, leading to a cascade of damaging events. When these autoantibodies bind to neuronal surface proteins, they can disrupt normal neuronal function by blocking receptors or altering signaling pathways. For example, autoantibodies against NMDAR can inhibit the receptor's function, leading to a reduction in synaptic activity and contributing to cognitive deficits and memory impairment. In addition to directly interfering with neuronal function, these autoantibodies can also trigger inflammation by activating the complement system, a group of proteins that form part of the immune response. The activation of the complement system can lead to the formation of the membrane attack complex MAC, which can puncture cell membranes and lead to cell death. This immune-mediated damage to neurons and synapses is a hallmark of ALE and was observed in the feline model through histopathological examination of brain tissue. Involvement of immune cells. In addition to autoantibodies, immune cells play a significant role in the pathogenesis of ALE. The feline model provided insights into the types of immune cells involved and how they contribute to the inflammatory process in the brain. Lymphocyte infiltration. The presence of lymphocytes, particularly T cells, was a prominent feature in the brain tissue of affected cats. These lymphocytes were often found in close proximity to inflamed areas of the limbic system, particularly around blood vessels, perivascular cuffing, indicating that they had crossed the blood-brain barrier to enter the brain. This infiltration of T cells is a key aspect of the immune response in ALE, as these cells can recognize and attack neurons that express the target antigens. T cells contribute to the disease process in several ways. They can directly kill neurons through mechanisms such as the release of cytotoxic molecules like perforin and granzyme. They can also produce pro-inflammatory cytokines, such as interferon gamma, IF and Greek letter gamma, and tumor necrosis factor alpha, TNF Greek letter alpha, which exacerbate inflammation and recruit other immune cells to the site of injury. The involvement of T cells in the feline model mirrors findings in human ALE, where T cell-mediated damage is a critical component of the disease. B cells and plasma cells, B cells, which are responsible for producing antibodies, were also present in the inflamed brain tissue. In the feline model, researchers observed an increase in B cells and plasma cells, the antibody-secreting form of B cells, within the brain, particularly in areas with significant neuronal damage. The presence of these cells indicates ongoing local production of autoantibodies within the brain, which can contribute to the chronic nature of the disease. The role of B cells in ALE is particularly important because it suggests that therapies targeting B cells, or the antibodies they produce, could be effective in treating the disease. In human ALE, treatments such as rituximab, which depletes B cells, have been used with some success, and the findings in the feline model support the potential efficacy of such approaches. Cytokine profile and immune activation. The cytokine profile observed in the feline model provided further insights into the immune mechanisms driving ALE. Cytokines are signaling molecules that mediate communication between immune cells and play a crucial role in regulating the immune response. Pro-inflammatory cytokines. In the feline model, there was an upregulation of pro-inflammatory cytokines in the brain and CSF. These included cytokines such as IF and Greek letter gamma, TNF Greek letter alpha, and interleukin-6, IL-6, all of which are known to contribute to inflammation and tissue damage. The elevated levels of these cytokines are consistent with an ongoing inflammatory response and are likely to contribute to the symptoms observed in the affected cats. The presence of these pro-inflammatory cytokines also suggests that they could be potential targets for therapeutic intervention. In human ALE, treatments that reduce cytokine levels or block their effects, such as corticosteroids or cytokine inhibitors, have been used to reduce inflammation and improve symptoms. The cytokine profile observed in the feline model supports the idea that similar treatments could be effective in this model as well. Immune activation markers. In addition to cytokines, the feline model showed increased levels of markers associated with immune activation, such as major histocompatibility complex MHC molecules and costimulatory molecules on immune cells. These markers indicate that the immune system is actively engaged in responding to perceived threats within the brain, driving the inflammatory process. The upregulation of these markers is a sign of a robust immune response and further confirms the autoimmune nature of the disease in the feline model. Implications for human medicine. The immunological findings from the feline model of ALE have several important implications for human medicine. First, the identification of specific autoantibodies and their role in the disease process provides a direct link between the feline model and human ALE. This link validates the use of the feline model for studying the mechanisms of the disease and for testing potential treatments that could be applied to human patients. Second, the involvement of both T cells and B cells in the inflammatory process highlights the complex nature of the immune response in ALE. This complexity suggests that effective treatment strategies will likely need to target multiple aspects of the immune system, including autoantibody production, T-cell activity, and cytokine signaling. Finally, the cytokine profile observed in the feline model provides potential therapeutic targets that could be explored in both veterinary and human medicine. Treatments that reduce inflammation by targeting specific cytokines or immune activation pathways could be developed and tested in the feline model before being translated to human clinical trials. The immunological findings from the feline model of ALE offer valuable insights into the disease's pathogenesis and provide a strong foundation for developing targeted therapies. Implications for human medicine. The research conducted on the feline model of autoimmune limbic encephalitis, ALE, holds significant implications for understanding, diagnosing, and treating the disease in humans. The findings from this model not only provide insights into the pathophysiological mechanisms underlying ALE, but also offer potential avenues for developing targeted therapies that could be beneficial for human patients. Advancing understanding of ALE pathogenesis. The detailed examination of clinical symptoms, pathology, and immunological responses in the feline model has advanced our understanding of ALE's pathogenesis. This model has highlighted the key role of the immune system, particularly autoantibodies and T-cell-mediated inflammation, in driving the disease. The similarities between the feline model and human ALE suggest that the mechanisms identified in cats are likely relevant to humans, making this model a powerful tool for studying the disease. Autoantibody-mediated damage. The identification of specific autoantibodies in the feline model that target neuronal surface antigens parallels findings in human ALE. These autoantibodies contribute to neuronal dysfunction and damage, leading to the characteristic symptoms of ALE, such as seizures, memory impairment, and behavioral changes. Understanding how these autoantibodies develop and cause damage is crucial for developing treatments that can prevent or mitigate their effects. T-cell involvement. The role of T-cells in the feline model has provided insights into how the immune system's cellular components contribute to brain inflammation. The infiltration of T-cells into the brain, along with their production of pro-inflammatory cytokines, suggests that T-cell activity is a critical factor in the disease's progression. Targeting T-cell-mediated inflammation could be a therapeutic strategy to reduce brain damage and improve outcomes in patients with ALE. Cytokine profile and inflammation. The cytokine profile observed in the feline model, characterized by elevated levels of pro-inflammatory cytokines such as IF and Greek letter gamma, TNF Greek letter alpha, and IL-6, mirrors the inflammatory response seen in human ALE. These cytokines play a central role in driving the immune response and causing tissue damage. Therapeutic strategies aimed at reducing cytokine levels or blocking their effects could be effective in controlling inflammation and preventing disease progression. Implications for diagnosis. The findings from the feline model have potential implications for the diagnosis of ALE in humans. The presence of specific autoantibodies in the blood and CSF of affected cats suggests that similar biomarkers could be used for diagnosing ALE in human patients. Autoantibody testing has already become a key component of diagnosing autoimmune encephalitis in humans, and the feline model supports the continued use and refinement of these diagnostic tools. Biomarker development. The identification of autoantibodies targeting specific neuronal antigens in the feline model could lead to the development of more accurate and specific biomarkers for ALE. These biomarkers could be used not only for diagnosis, but also for monitoring disease activity in response to treatment. For example, measuring autoantibody levels in patients over time could help clinicians assess the effectiveness of immunotherapy and make adjustments as needed. Early diagnosis. The clinical symptoms observed in the feline model, such as seizures and behavioral changes, are often the first signs of ALE. Recognizing these early symptoms and testing for autoantibodies could allow for earlier diagnosis and intervention in human patients. Early diagnosis is critical for preventing irreversible brain damage and improving long-term outcomes. Therapeutic implications. The therapeutic implications of the research on the feline model of ALE are profound. The findings suggest several potential treatment strategies that could be explored in human clinical trials. These strategies focus on targeting the immune system to reduce inflammation, prevent neuronal damage, and improve neurological function. Immunotherapy. The success of immunotherapies in treating other autoimmune diseases suggests that similar approaches could be effective for ALE. Treatments that target B cells, such as rituximab or reduce autoantibody production, could help control the disease. Additionally, therapies that inhibit T-cell activity or block the effects of pro-inflammatory cytokines could reduce brain inflammation and prevent further damage. The feline model provides a valuable platform for testing these therapies before they are applied to human patients. Anti-cytokine therapy. Given the elevated levels of pro-inflammatory cytokines in the feline model, anti-cytokine therapies could be a promising approach for treating ALE. Drugs that block the action of specific cytokines, such as TNF Greek letter alpha inhibitors or IL-6 inhibitors, have shown efficacy in other inflammatory diseases and could be repurposed for ALE. The feline model offers a means to evaluate the safety and efficacy of these treatments in a controlled setting before moving to human trials. Neuroprotective strategies. In addition to targeting the immune response, strategies aimed at protecting neurons from damage could be beneficial for ALE patients. Neuroprotective agents that prevent neuronal death, reduce oxidative stress, or promote synaptic repair could help preserve brain function and improve cognitive outcomes. The feline model could be used to test these neuroprotective strategies, providing a preclinical basis for human studies. Broader implications for autoimmune neurology. The implications of the feline model extend beyond ALE to other autoimmune neurological disorders. The findings from this model could inform research on a range of diseases where the immune system attacks the nervous system, such as multiple sclerosis, neuromyelitis optica, and other forms of autoimmune encephalitis. The insights gained from studying ALE in cats could be applied to these conditions, leading to a better understanding of autoimmune mechanisms in the brain and the development of new treatments. Model for other diseases. The feline model of ALE could serve as a template for developing animal models of other autoimmune neurological disorders. By adapting the methods used in this study, researchers could create models for diseases that currently lack good animal representations. These models would be invaluable for studying disease mechanisms, testing therapies, and developing diagnostic tools. Cross-species insights. The similarities between the feline model and human ALE underscore the importance of cross-species research in understanding autoimmune diseases. By studying ALE in cats, researchers can gain insights that are directly applicable to humans, demonstrating the value of veterinary research in advancing human medicine. This cross-species approach could be extended to other areas of neurology, where animal models can provide critical information that is difficult or impossible to obtain in humans. Conclusion and future directions. The research conducted on the feline model of autoimmune limbic encephalitis represents a significant advancement in our understanding of the disease. The findings from this model have provided detailed insights into the pathogenesis, immunology, and potential treatment strategies for ALE, with direct implications for human medicine. The similarities between the feline model and human ALE validate the use of this model for further research and underscore its potential as a platform for developing new therapies. Future research. Future research should focus on refining the feline model to better understand the molecular mechanisms underlying ALE and to identify additional therapeutic targets. Longitudinal studies that follow the progression of the disease in cats could provide valuable information on the natural history of ALE and the factors that influence its outcome. Additionally, the model could be used to test new treatments, both immunotherapies and neuroprotective agents, to determine their safety and efficacy before moving to human clinical trials. Translational potential. The ultimate goal of this research is to translate the findings from the feline model into effective treatments for human patients with ALE. By continuing to explore the immunological and pathological mechanisms of the disease in this model, researchers can develop new therapies that are grounded in a deep understanding of the disease process. The insights gained from the feline model could lead to breakthroughs in the diagnosis and treatment of ALE, improving the lives of patients affected by this devastating disease. In conclusion, the feline model of autoimmune limbic encephalitis provides a detailed and accurate representation of the disease, offering valuable insights that are directly applicable to human medicine. The findings from this model have the potential to advance our understanding of ALE, improve diagnostic and therapeutic strategies, and contribute to the broader field of autoimmune neurology. This concludes the expanded version of the article. The entire content now provides a comprehensive overview of the clinical presentation, pathology, immunology, and implications for human medicine based on the feline model of ALE.